Bon, ben merci à tous d'être venus. Thank you everybody for coming to today. So today we have a visitor from University of Queensland in Australia. So Sam Robinson, who is collaborating with Axel Touchard. You want to say something? No, okay. And yes, he will be presenting his work on ant venoms. So a very exciting subject indeed. Okay, well, Sam,
Merci. Okay. Try not to fall over. Thank you for having me. As you'll see, my presentation today will be in English, so I apologize beforehand. My French is not too good. And today I'll talk about sort of my research program in general at the University of Queensland, which I can sort of sum up as using venoms to identify new pain pathways. Oh, hang on, is it on or it's off? Yeah, okay. Okay, so a bit of background about me. I'm actually from New Zealand originally. I did my Bachelor of Science there and honors there. And then I worked as a research technician in molecular neuroscience, studying these neurons here. In 2011, I decided to do a PhD. I chose a subject in toxinology. So that's the study of toxins. And in order to do that, I needed to move to Australia. And I ended up at a place called MIPS, or Monash Institute of Pharmaceutical Science, down in Melbourne in Australia. And after 5 years there, oh, I should point out the snail is there because that's what I worked on during my PhD, the venom of these sea snails called cone snails. Then I moved to the sort of leading lab in our field of toxinology, and that's at Toto Oliveira's lab at the University of Utah. So there I worked on cone snails again, but many more cone snails. I didn't just have one species. And I was able to tie in some work that we're using an assay, which I'll talk about later, where we were looking at sensory neurons. And we were using that assay there to try and identify targets for toxins from these venoms. And then I moved back to Australia in 2017 to the University of Queensland, and I needed to come up with my own research program. And for that, I put together my my background experience in neuroscience and my work with venomous animals. And I needed to stop working on cone snails and do something else. So as you'll see, I'll show you, I've branched out into other stinging things. So my research program is sort of based around some very simple observations, if you like. The first of these is that venomous animals, almost all of them will sting defensively. And that's something probably most of us have experienced, I would imagine. And the hypothesis I make from that is that defensive toxins must therefore exist. The second observation is that many stings cause immediate, sometimes intense pain. And again, most of you may have experienced that, but perhaps you haven't thought about it beyond that. So what's going on when you're stung is that there's a chemical in that venom that's basically targeting your sensory nerves and sending a signal up to your spinal cord and then up to your brain where you experience the pain. And most people will then withdraw from the pain. Your brain will tell you to get yourself out of that situation. So this is an adaptive system that we have to protect us, right? It's good that we can feel pain. If we don't feel pain, we usually die quite young. And it helps us, we can detect something like a fire and avoid it, things like that. These insects or different stinging things, what they're doing is exploiting that system that we have. And they've got chemicals that basically target that system or override it to cause pain. They're essentially tricking or deceiving our nervous system. And that's my second hypothesis, that these venoms contain toxins that target our sensory nervous system. And the third observation that perhaps less people are familiar with is that not all stings are equal. So a sting from a certain wasp may be quite different to a sting from another wasp or ant. And this picture I'm showing here on the right is a really nice illustration of that. So this illustrates something called Schmidt's sting pain index, and this was Justin Schmidt is an entomologist from the USA. He basically worked for decades on stinging Hymenoptera, so bees, ants, and wasps, and he recorded what those stings felt like. He just was stung as he went about his work as part of his job, and he recorded that. And he came up with descriptions and also the level of pain that he experienced. So for example, the maximum pain is say level 4. And then the intermediate one is level 2, which is something like a honeybee, and your average sting from a honeybee. And then something that doesn't hurt very much might be a 1 or a 0.5 or something like that. And his descriptions of these things are something like this. So an example is he talks about sweat bees, and he describes the sting as light and ephemeral, almost fruity. A tiny spark has singed a single hair on your arm. And then he moves on to something like a Florida harvester ant, which he describes as bold and unrelenting. Somebody is using a power drill to excavate your ingrown toenail. So the point I'm trying to make here is that these are obviously quite different. And if you've experienced these two things, you know that the chemistry between them is different, and something different is happening to your body when you're stung. And I've sort of expanded Justin's index. He worked on bees, ants, and wasps, and I've expanded it to other things. If you're interested, you can check it out on my social media. But the sort of summary here I'm trying to make is that there's actually structurally and functionally diverse chemistry in these venoms, even just within the Hymenoptera. So they're probably a really good source of pain-causing toxins, and that's using that information is how is sort of the goal of my research. So we know that venoms, or anyone in toxinology, perhaps not everyone here, but we know that venoms have been, or toxins from venoms have been really, really useful to help us understand the physiology of the cardiovascular system and also the neuromuscular junction. And that's because there's toxins that have evolved in, say, a snake venom to specifically target certain components of these systems. And they've been really helpful over the last few decades. But no one's bothered to look at things that cause pain because there's no immediate application, right? But I figure if we can use it to study these systems, why not use it to study the sensory nervous system, particularly given that these things have presumably evolved to target components of the system. And if we do that, perhaps we can identify new pain targets, so different ion channels or receptors on our sensory nerves. And ultimately, that might lead to new treatments for different types of pain. So the first question was, back when I started this, was where do you begin? And if you look at this tree here, this is all the tree of life, and you see everywhere there's color, that means these are venomous things. So there's a lot of venomous things out there. In fact, venom's supposed to have evolved more than 100 times independently, which I think sort of astonishes a lot of people. So there's this huge diversity out there. My question was, where do I start? And really it was sort of whatever I wanted at the time. And I was most interested in Hymenoptera because that was what I was familiar with. So bees, ants, and wasps. And I started with the ants. And since then, this has led to some really good collaborations, including Axel, who's in the room here, and also others. And from that, I was able to build up a skill set that allowed me to answer these questions, and other people noticed this. And I was able to form some collaborations both in Australia, so working on assassin bugs, stinging trees and caterpillars, stinging caterpillars, but also overseas. So I collaborated with Nick Casewell in Liverpool in the UK to look at the pain-causing components in spitting cobra venom. And I've also started expanding this work to look at these and other Hymenopteran venoms. And this is also partly why I'm here working with Axel. So today I'm just going to talk about the work that I've done with ants rather than all of that. So the first ant I worked on was this what we call bull ants in Australia, or the genus Formicidae. It's actually a family subfamily that's mostly endemic to Australia. There's about 100 species. We have different names: bulldog ants, inch ants, jumper ants. Inch ants is because they're about an inch long. Some of them, they can be quite big. They're common, they're aggressive, and they have a very painful sting. So they're perfect candidates for my research. Our first job was to try and figure out how to actually collect venom from these ants. And this is something probably about the same time Axel was working on independently. And this is what we came up with. You can stretch some parafilm over a tube and just allow the ant to do what it naturally does, and that is to sting aggressively. And what you can actually see there is, if you look closely, little drops of venom collecting on the surface of that, and we can just rinse those off, and we have this pure venom that we can then work with. That approach doesn't always go right, and sometimes the ant gets off, and I have been stung more than once doing that, but it's effective. So by doing that, we can collect venom. We run the venom on high resolution mass spectrometry instruments, so we separate it out, and that's what's shown on the right here. Is each peak, you're separating out that complex mixture, and each peak represents a different component in that mixture. And then what we want to do is assign each of those peaks. So try and figure out chemically what those are. And to do that, we use transcriptomics. So we take the venom producing organ. So in ants, this is this venom gland pictured here. And that's responsible. It's like a factory for producing these toxins. It has all of the RNA that encodes these toxins, and you can use that, build a transcriptome library, and basically, without going into too much detail, you use that as a database to basically figure out what each of these components are. And we've got that down to quite a fine art, and we can figure out exactly what's in these ants. And so at the time, this is a few years ago, and it's still a bit common now, is that people think of ant venoms as being just formic acid. And people who work on ant venoms, for example, myself or Axel, know that this is absolutely not the case. But it was nice to sort of show that. And I think at the same time, Axel published a few papers showing the same thing, that they're in fact peptidic. And we identified a total of 15 peptides, toxins in this venom. Now, I don't need you to understand this slide. It's a bit intimidating, but at the time we were really excited because it was one of the first sort of look at a complete ant venom, what was in it. And prior to that, we'd only seen little isolated snapshots of what were in these venoms. As you can see here, these are the peptide sequences. And without going into any detail, basically the ones I've boxed here in red, they're all related. They come from the same gene superfamily probably. And they're all sort of biochemically similar. And basically you can sort of sum them up. We called this family of toxins the Aculia toxins, after the Aculia Hymenoptera. Typically they're cysteine free, or they don't have a lot of cysteines. The sequences are very hydrophobic, or the amino acids in it. But they're also cationic, and what that means is, and they're spaced accordingly, so that when those peptides make an alpha helix, all of the hydrophobic residues are on one face and the cationic residues are on the other, and that makes them what we call amphipathic. And this sort of structure is typically associated with membrane activity. So like a detergent, they'll get into a membrane and disrupt it. And this is what, if you do that in a neuron, you'll form a pore, you'll depolarize the membrane, you get an action potential, and that'll lead to nociception. And that's exactly how melatonin works, which is the major component in honeybee venom. And what we found was that actually there's a homologous thing in the bull ant venom that's doing basically the same thing as melatonin, as far as we can tell. So if you're following, that result wasn't, it was interesting. We got to have a close look at an ant venom, but it didn't help us identify any new pain targets or receptors. Yeah. Nociception is pain. Yeah. We're just not allowed to call it pain because pain is something humans experience. It's got an emotional component. Nociception is the physical component of pain. Yeah. I get told off by reviewers all the time for calling it pain. Yeah. All right, so it didn't tell us anything new about pain targets. We didn't identify a new receptor or ion channel that was being targeted by this venom. So in that sense, it was a little disappointing. But myself, people like Axel and others knew that there was more to Hymenoptera venoms than just these membrane targeting peptides. And this is, I mean, it's clearly illustrated in this simple figure, is that there are these things like the bullet ant pictured in the center that have much more painful stings than, say, other stuff like a honeybee. And they also have some neurotoxic effects, which are exploited in some indigenous rituals. So we asked the question: Is there more to membranes than the toxins in ant venoms? And there's some evidence from a few decades ago where Tom Pieck and his colleagues identified what they called a neurotoxin, showed that it affected sodium currents in frog muscles, which was just the system they were using to study these toxins. And then actually a while later, Steve Johnson, quite recently, 2017, or recently at the time when we started this, showed that actually this toxin or this venom could affect what we call sodium channels. And another question was, is there more than just the bullet ant? So we have this ant in Australia called the greenhead ant, or Rhytidopinera metallica. It's really common. So if you have a grass lawn, you basically have this ant, and people say that you think you own the lawn, but actually the ant owns the lawn. So you can't use it unless the ant lets you, typically. And they're really widespread in Australia, including in suburbia. So these stings from these also cause severe, long-lasting pain and some sort of kind of strange neurotoxic symptoms. So what these words mean is they basically cause goosebumps and sweating at the sting site. So again, it alluded to something a little more complicated going on there. So there's something special about certain ant stings. So our aim was to try and figure out what the responsible toxins were and how they were working for these two ant species. And this is where I was working with Axel already on figuring out what was in bullet ant venom through a mutual collaboration. And we showed that actually this venom is incredibly simple. So when you separate out what might be a complex mixture, you actually basically see one peak. And when you look at the transcriptome, it's this peptide we call ponerotoxin, or PC1A, that makes up the vast majority of that venom. So our answer was pretty simple. We made that peptide, tested it. And what we do here is we inject that into the paw of a mouse. So we're trying to mimic like a stinging event. And then we record it, and we basically, someone who's blinded to it records whether there's pain behavior events. So the maximum pain behavior you can get in this assay is 300, so events per 5 minutes. So you can see that basically you inject ponerotoxin, you go straight up to maximum, and you stay at maximum. So we had our candidate for what that pain-causing toxin was. That was pretty easy, from ponerotoxin, from Paraponera venom. When we went to the greenhead ant, it was a little more complicated. As you'll see, this is the equivalent trace here on the left of a run under basically the same conditions. You can see there's a lot more peaks. So there's a lot more going on with this venom. And when we do the same thing, we make a transcriptome, we actually found that there's over 100 different peptides in there. So it was going to be a lot more complicated. But fortunately, we could group these peptides into different groups. And what I did was make a representative of each group, repeated the assay where we injected into mice, And there was one really obvious candidate that got close to maximum pain, this peptide we call RM4A. So that was our candidate from the green ant. And about the same time as where Axel emailed me and he said, oh, I've got this really, really interesting ant that he'd gone all the way to Cameroon to collect. And he told me that he considered the sting to be more painful actually than the bullet ant. And could we figure out what the component was? So we started that collaboration too. And he sent me some venom. I separated on HPLC, and you see each of these different components. So it's quite a complex venom. And then we got here, what I did was I tested each fraction on these cells we call F11 cells, which are just sensory neurons from mice. So if we see a signal, that means those cells are activated, and that means that toxin is probably going to cause pain. And here we saw a signal coming from this peak of this venom component and these two little peaks here. And I said to Axel, oh, it's probably boring, you know, it's probably just this peak. This is just some artifact. We'll ignore these. And he was very adamant that no, he wanted to make these ones as well. And he wanted to test these. Unfortunately, he stuck to it because we made them, and we found that actually it was these that are the pain-causing component in the venom, and they're about 2,000 times more potent than this one. So that's what's shown there. And that was this peptide we're calling TA3A. And some of you might have realized that if we look at that compared to RM4A from the green ant, it's almost the same thing. So they're homologous. So we've got 3 ants that are using apparently something pretty similar to cause pain. The next step was to figure out how they work. So what ion channel or receptor are they targeting in our sensory nerves in order to send that pain signal? And the assay I've sort of developed to test this is this one here. So I'll just explain it briefly. Basically, DRG stands for dorsal root ganglia. So these are these ganglia close to your spine where all the cells are that send out their neurites to your periphery. And when you experience something dangerous or a stimulation here, it sends the signal up that axon, and that's how you experience pain. So these are your pain sensing neurons, your DRG neurons. And I can take these out of a mouse and culture them. And then I can load them with a dye that allows me to see when they're activated. And because they're coming from a mouse, they're not just like a cell line where every cell is the same. Actually, you've got different sensory neurons that sense different things. For example, some sense heat, some sense painful things, some sense acid. And that's because they have different ion channels and receptors on them. They all have a different job. And that's one of the strengths of this assay is you can look at this heterogeneous population of neurons all at the same time. So under the microscope, when you look at it, you see something like this. So these are all DRG neurons in culture. They're loaded with a calcium sensitive dye. And then I add a toxin or a venom to it, and you see the cells light up. And that tells you that those cells have been activated by the venom. And then at the end, I add a positive control, and that shows me all the neurons in there. And what I can do is look at background and toxin activated cells. I can look at every individual cell as it's activated or not activated, and I can look at how those fit in with the overall cell population. So which subpopulations of cells are being activated, and it's an incredibly powerful technique for telling you what your target might be. So when we put Pinerotoxin on there, it basically activated every neuron. So you see 90% of neurons were activated. So it's directly activating our pain sensory neurons. And what we can do is add a different blockers in there. And what I found was if you add TTX or tetrodotoxin, which is a blocker of voltage gated sodium channels and ion channel in our nerves, you can suppress that completely. So what that indicates that Pinerotoxin directly activates our sensory neurons, and the receptor for that toxin is TTX sensitive. In other words, it's probably a sodium channel. And here, this might look a bit intimidating if you haven't done electrophysiology, but just bear with me. It's not too complicated. So basically, if you've worked with ion channels or receptors, you know that they have an effect. The effect that you get for an ion channel, like a sodium channel, as the name suggests, is that sodium flows through a channel. The channel opens, sodium ions flow through, and we can measure that. And that's what this little black spike indicates. That means the channel's been activated. There's a little spike in current as the ions flow through, and then it inactivates. And you can cause that current by making a voltage step. So voltage, you view voltage as the agonist or the ligand of these channels. They're what activates it. So if you step from minus 90 millivolts to minus 20, that will cause the spike and current to go through. And that's why the channels are called voltage gated, because they're gated by a voltage stimulus. When you have the toxin present, toxin, it activates as normal, but then it doesn't inactivate fully. So you've still got all this current flowing through. And actually what we observe is that it works at different subtypes of these sodium channels. I should point out NaV17 is the one that we know is really involved in pain. If you knock this out in humans, you can't experience pain anymore. So it makes sense, but it's also even more potent at NaV16. And the other thing it does is a channel will usually open, say, at minus 30 millivolts. That's when it'll reach its threshold and ions will start to flow through. When you have the toxin present, it opens much earlier. So what you have is this channel that's really, really sensitive. The toxin makes the channel really sensitive. It makes it open much earlier, and it allows a lot more current through. And that's having that extra current through a sodium channel in sensory neuron is what causes that pain we experience when we're stoned. When we tested RM4A, we basically saw exactly the same thing. So the green head ant is doing the same thing as the bullet ant. And when we tested TA3, it was similar, but there were just a little difference. So this is the toxin from Tetramorium africanum venom. You'll see the one difference here is that instead of when we repolarize, you'd usually go back to baseline, but here the current just keeps on going through. So this will actually eventually kill the neurons, and so it causes pain, and then the neurons die. So it's a pretty nasty toxin. It's a very, very interesting toxin. They all are. So what we found is that actually the key points here is that it's peptide toxins that are causing the pain. We didn't know that before. Certainly not formic acid. They directly activate our sensory neurons, and they target voltage-gated sodium channels to do this. They're actually unique among described toxins. They're a new class of toxin. And I've put the little picture of the frog here because the effects they have on sodium channels are actually very closely related to a toxin called batrachotoxin, which comes from a poison dart frog. So there's this, these toxins have convergently evolved to target the same thing. This has told us how ant stings hurt, but it's also given us a few new tools to study sodium channel function in the context of pain. So that's the first part of my talk. I'll quickly go through another story that involves, I'll go back to Myrmecia. So you remember I showed this slide, which you probably thought was quite boring, just a list of peptide sequences. And I said that these were all related, but there's one at the bottom that's different. And this was a little bit of a puzzle to us. And I had an honors student starting, and he wanted a project. And I said, oh, why don't you figure out what this toxin is doing in there? So we looked at it in the venom. It's this peak here, and actually it's one of the most highly expressed things in the venom. When you separate the venom out, this peak is the biggest actually in this view. So there's lots of it there, and it's highly expressed. This is the counts in the transcriptome. It's right up there with the other toxin. So it's a major venom component in the bull ant. Okay. Cool. There we go. Okay. So I said it was different. I'll explain that a little bit here. So if this is the sequence, the amino acid sequence of the toxin, and you'll see there's a lot of cysteines in there. It's not hydrophobic, it's not cationic, it's not one of these other toxins. And you show this to a biochemist, they'll say, oh, it's going to be a neatly folded peptide. And some will even say, well, it's what you call an EGF fold. So it's this epidermal growth factor fold, which is reasonably common. And we have it in a suite of hormones that we have in our body, EGF peptides, or have this fold. And those are actually what I've aligned this sequence with here. So called, say, for example, HBEGF, epiregulin, beta cellin. These are all human hormones, or mammalian hormones, I should say. So my honors student at the time, David, I said, go and solve the structure, prove to me that it's an EGF fold. And he did that. And you can see here, maybe you're not familiar with it, but I'll tell you that's an epidermal growth factor fold. So he showed that. And then the next question I told him to answer was, our hypothesis was this toxin, it can either be in the venom to cause pain in defense, or it can be there to, when the ant stings its prey, it might be there to paralyze the prey. These are really the two main options. So in order to test that, he injected into the mice the same way I explained before, just an injection into the paw, and he saw nothing. So we thought, oh, well, it's not involved in defense. And then he injected it into a couple of different types of insects to see if it paralyzed them or killed them. And again, it did nothing. So it was a little bit disappointing for him. And it was about that point that his honors year finished. So it was a bit of a shame. But he stayed on and did a PhD. And I said to him, like, I said, if you want it to be a good paper, you have to work on it during your PhD as well. And he credit to him, he did. And during actually, this was about when COVID hit, and I was stuck at home on quarantine, and I thought I'll revisit this study. It was sort of going nowhere, and I'll just have a look through some of the data. And so I took the amino acid sequence and I blasted it. And I think probably most of you are familiar with BLAST, just you align it to a database of known proteins. And I'd done this before, but I hadn't really looked at it in detail. And what I noticed this time was that actually every single hit in this huge database, there's a lot of EGF peptides in this database, every single hit, all 100 of the top hits were to vertebrate sequences. So things that were not related to ants, sequences coming from vertebrates, even though ants have these EGF domain peptides. And I thought that's a bit unusual. And then it got even weirder. So I looked at the top 3 hits, and actually they all come from Australian endemic mammals, which are the koala, the wallaby, and the wombat. And again, this was kind of weird. I thought, this can't be coincidence. We have this ant that's endemic to Australia, and it's got this toxin that resembles hormones from these Australian mammals. And actually, when I dug a bit deeper, you can see this alignment of the sequences here. This is the toxin from the ant. And actually, there's a sequence from a mammal that it's 60% similar to, whereas the closest insect sequence is only 33% similarity. And again, the disulfide spacing and thing is consistent with a mammalian sequence rather than an insect sequence. So it's like this ant somehow had a mammalian hormone in its venom. And it got even cooler. So the top hit we found was actually to the short-beaked echidna, which is also known as the Australian anteater. So it's the one animal in Australia that will actually dig up the colonies of these Formicidae. Everything else leaves them alone. So again, we have this peptide toxin in this venom that the closest thing to it is actually a hormone of this mammal. And again, we sort of thought, well, this is kind of weird. And we tried a few different things. We didn't just rely on the sequence alignments. We made a PCA plot based on the physicochemical properties of the toxin or the peptides. And again, we saw that the toxin grouped with vertebrate sequences, not with the invertebrate ones. when we aligned searched the structure against the database, we found that the structure aligned with vertebrate hormones, not with the insect peptide again. So it really was functionally and structurally analogous to a vertebrate hormone, not to an insect peptide. So I said to David, go and test it, see if it's active on the mammalian receptor for these hormones. So this is the ERB receptors. Don't ask me what that acronym stands for. I actually don't know. But they're basically the EGF receptor. So he used a couple of assays. He showed that indeed they were active there. In fact, the peptide toxin was more potent than EGF, the endogenous human hormone, at these receptors. So yeah, it was pretty clear that we had an analog of a mammalian hormone, something that looked like that rather than like a toxin. And again, we were kind of stuck. Because we didn't have any data showing what the function might be. Again, it was inactive when we injected into mice, as far as we could tell, and it did nothing in insects. So we went back to doing some reading to figure out what these hormones do in humans, and we found some literature showing that one of them, it's a major component of wound fluid. So when you have a blister, it's in there and it's helping to heal the wound. We found some literature showing that if you take these inhibitors, which some people do for certain types of cancer, that can lead to reduced pain. So the reverse logic is that perhaps if you take the agonist, it can actually cause pain. And then the other thing that came out later was actually the signaling was linked to chronic pain through whole genome studies. Again, we were sort of stuck because we'd done the assays that we could do and we didn't know where to go. And then a colleague knew that I was sort of puzzling over this and sent me this paper that when it came out in March 2021, which is a while ago now, and said, oh, this EGF peptides are now linked to hypersensitivity to pain. And so what they'd done in this study is, instead of measuring direct spontaneous pain, they'd weighted and they'd measured whether the mouse was sensitive to a mechanical stimulus. So all you're doing there is you inject the thing, you wait for a bit, and then you apply a mechanical stimulus, like a little wire, to the mouse's foot, and you measure the threshold at which the mouse feels that. And basically you can see whether mouses become more sensitive to that mechanical stimulus. And what they'd showed was that this human hormone, if you inject it into mice, actually caused that sensitivity. And I thought, voila, I just go and test it on the same assay, and maybe we'll see the same thing. So we did that, and again, remember, it was not active spontaneous pain. And then we tested it on measuring the hypersensitivity test, and actually the peptide made the mice much more hypersensitive to just a normal mechanical stimulus. So we found the function of this peptide essentially. And I think probably the coolest thing is that, sure, we showed that it's long-lasting hypersensitivity this peptide causes. It's a new defensive mechanism of action. It hasn't been described before for a toxin or a venom. But I think the coolest thing is the mimicry. So the fact that this ant has evolved a peptide that mimics so closely a hormone in a mammal. And the only other type of the specific type of mimicry that's been described comes from Heliconius and Passiflora, so butterflies and passion flower vine. And what you see here is these little nodules. They look a lot like butterfly eggs, but they're actually grown by the plant itself. The plant has evolved to grow these little things. And what that does is the butterfly then avoids laying its eggs on this leaf to avoid competition with other eggs. So it's a really clever way of the plant avoiding having this butterfly lay its eggs on the leaf. And the bull ant and the echidna, something analogous is happening. The bull ant has evolved a hormone or toxin that resembles something in the echidna in order to deter it. And it's a really cool example of natural selection shaping a gene in one organism to resemble another, or molecular mimicry. And the other thing it's told us is that perhaps the EGF receptor is worth targeting as a pain pathway. There's not much literature on that. It's actually just started emerging recently that it might be worthwhile. And it tells us that EGF receptor signaling is probably involved in pain. It probably has an adaptive function, but our question is, can it also be pathological? So can EGF receptor signaling go bad, and is it worth suppressing? And these are all questions that this study of an ant venom has now raised. So yeah, I'll sort of finish up there. I'll say I've given you two examples of where we've studied ant venoms, but actually there's a whole lot more venomous things, almost an endless source of stinging things to look at, and a library of pain-causing toxins that I'll continue to look through. It leaves me to thank those involved in the study, particularly Axel for his contribution, which was really critical. And thank you all for listening.
Thank you, Sam, for a fascinating presentation. It was great, awesome. So could I ask the people that are online if they have any questions? Okay, so then I'll ask the room. Has anybody got any questions, or vous avez des questions? Allez.
Thank you, Sam, for your great presentations. So do you think that some of this pain-inducing peptide could be turned into painkillers?
That's not really the goal of what I'm trying to do. So my interest is in understanding the pain-causing chemistry. But yes, I do think it is possible. So sometimes a activator, into a peptide activator, can be structurally very, very similar to something that's a blocker. So you can make very subtle changes to a peptide to take it from being something that causes pain to something that potentially blocks pain. So yeah, there's definitely potential. I'm not looking into it myself, but I think this will lay a foundation for others to do that. Yeah. Yeah, it's an interesting question. One thing I want to look into is some Hymenoptera have evolved to only use their venom defensively. So a lot of the ants we work on will use it for prey capture and also for defense. But some Hymenoptera, say wasps, eusocial wasps, will use the venom also just for defense. So we can look at the difference between a wasp's venom that uses it just for defense and one that uses it for both defense and predation. Yeah. Yeah, I have actually tested that. So for example, these ones I showed here, you could have the two hypotheses that these toxins are there, they've evolved just to cause pain defensively in vertebrates, or it could just be a secondary effect. They could actually be a toxin. They could have evolved to paralyze insects or have an insecticidal effect. But this is just a side effect that they cause pain. So we took those three peptides. Actually, Axel did this himself, injected them into flies to see if they were insecticidal. And I think you needed to increase the dose by about 20,000-fold to make them insecticidal compared to what causes pain in insects. And one of the peptides, no matter how high the dose was increased, it was inactive. So I think that was about as strong evidence as we'll get that these particular toxins, they have evolved as defensive toxins to target vertebrates rather than invertebrates. But it's a little more complex than that because these toxins had to come from somewhere. And I think my theory for most of these pain-causing toxins is that initially the precursor for them was in fact a predatory toxin. So the ant, perhaps, for example, had a predatory toxin. It then had a defensive selection pressure. It needed to defend itself. The ants that had a toxin that had a secondary effect to cause pain are the ones that survived. And then this toxin branched off and became a specific defensive toxin. Yeah.
And if we extend this to vertebrates like snakes or organisms that only use their venom as defensive, what would be the prediction or hypothesis?
So can you?
If you're a snake,
yeah,
your prey are mainly vertebrates.
Yes.
So you can there are confounding effects between inducing pains and preying. And if you are other kind of organism that use venom only as defense, like nettle tree, for example, from an evolutionary perspective, you should develop defensive toxins only, so not having any side effect.
Yeah. And actually, yes, that's exactly what I've seen. So I've done some work with spitting cobras in collaboration with Nick Casewell. And what we found was that the two main peptide components of the venom are these what we call three-finger toxins, because they look like three fingers, the structure does. You have ones that target the mammalian nicotinic receptor, the neuromuscular receptor, to cause paralysis. And there's a lot of those in the venom. But there's also ones that are cytotoxic. So they have a lot of, they have a structure that makes them cytotoxic. And in the spitting cobras, the ones that use their venom more defensively, actually, I've got this wrong. They both of them have each of these cytotoxic and so defensive pain-causing toxins and the neuromuscular targeting or predatory toxins. But the spitting cobras had an increased expression of a phospholipase enzyme. All of them did, which actually promoted the effect of the cytotoxins, so made them more effective. So in the case of those snakes, there's both present, and it doesn't seem, maybe there's some balancing thing going on. I'm not sure. In the case of the nettles, we only identify obviously a pain-causing toxin, right? It has no need for a predatory toxin that I'm aware of. Yeah.
But I think with the snakes, you can do the difference between the peptides that are just for defensive, in terms of the spitting cobras. Yep. There's a blinding your opponent. Huh? Yes. No, because I think that was part of the question, that in snakes you can't make the difference between predatory and defensive.
Oh, you can.
Yeah, with cobras you can. Okay. It was more of a comment in terms of your last example. It's absolutely fascinating. So basically, it's mimicking the gene. I find that absolutely gobsmacking. Yeah. It's a completely new way of seeing venom. It's basically not just attacking. Okay.
It's interesting. That's not the first time I've seen that mimicry. So when I was at the University of Utah, I was working with Helena Safavi, and she was working on these peptides from a cone snail venom, and it's actually it's a mimic of an insulin molecule. So the snail releases it into the water, and it causes the fish to be sedated, and then it can swallow the fish. And the insulin in the cone snail looks exactly like fish insulin. It doesn't look anything like mollusk insulin. So there's a few examples out there of this kind of molecular mimicry, and it's probably more common than we realize, I think.
All right. New way of seeing things. Est-ce qu'il y a d'autres questions? Okay. Well, thank you, Sam, so much for this amazing presentation. And thank you everybody for coming here.